Hello, everyone. It is my great pleasure to welcome you all to the third seminar of the Ocean Insights, Indian Ocean Seminar Series by Early Career Scientist Network of IIOE 2. Our speaker for today is Tanya Marshall. She did her PhD from the Department of Oceanography of the University of Cape Town, South Africa, and she has just started her postdoctoral research in the same institute. Her research interest is in the exploration of ocean biogeochemistry using nitrate isotope ratios, nutrient stoichiometry, geochemical tracers, and ocean physics. She is additionally involved in the exploration of nutrient fluxes, particularly nitrogen cycling in the South Atlantic Ocean and the Indian Ocean. Today, Tanya will present her work on nutrient fluxes in the Southwest Indian Ocean and its implications for regional fertility and carbon drawdown. With this very brief introduction, I now invite Tanya to please initiate her talk. Thank you.
Hi, everybody. Thank you so much. Let me share my presentation. Can you just confirm that you can see it and hear everything? Okay, great. I'm going to minimize the video. I can't see anybody, but just shout out to me if there's a problem. Okay. Hi. Thank you for the introduction. I'm really excited to be talking to the Indian Ocean community. So thank you for having me here. Today, I'm going to be sharing work in my PhD thesis, focusing today on nutrient fluxes in the Southwest Indian Ocean, mostly looking at nutrient supply mechanism to this oligotrophic surface ocean region. So some of this work is already published, and I'll indicate where that's the case, but some of it is still in review. So I've also indicated that, so please just be conscious of not sharing those figures or our findings yet. Hopefully it will be published soon. Okay, so I'm going to start with a framework. So we know in the low latitude surface ocean that nitrogen availability modulates ocean fertility and climate. So here I'm showing a schematic of the new production paradigm, where in the pink box, I'm showing regenerative production. So that is just recycling of nitrogen in the surface ocean and has no implications for carbon export. And in the purple box, that is new production. And that equates to, of appropriate timescales, export production. So I'm going to talk about new production today. I'm not really going to talk about regenerated. So I'm actually just going to focus on two aspects of new production, and that is N2 fixation and deep nitrate supply. Sorry, I have a bar here that's blocking my slides. I think that makes it easier for me. Okay, so two mechanisms of nutrient supply that fuel new production. So new production is phytoplankton growth fueled by nitrogen that is new to the euphotic zone. So N2 fixation is the ultimate supply of nitrogen to the ocean, and it results in carbon sequestration. So that's the orange box. So really, it reduces atmospheric CO2 concentrations by storing carbon in the deep ocean for long time scales. And then mixing of deep nitrate, right, in this blue box does contribute to fueling new production. So it does supply nitrogen that is new to the euphotic zone. However, when we upwell deep nitrate, we also upwell a stoichiometric quantity of DIC. What that means is that we upwell CO2 basically that can efflux in the surface, so such that when the phytoplankton consume this new nitrate, they also draw down DIC, so there is no net reduction of atmospheric CO2. But carbon export is important for maintaining a vertical gradient in DIC, so basically maintaining the soft tissue pump. So both aspects are important, but N2 fixation, does result in the lowering of atmospheric CO2 concentration. So that's the difference. Oops, sorry, my slide isn't changing. Okay, so here I'm showing a global map where the distribution is a modeled prediction of N2 fixation. And generally what you see is that N2 fixation is predicted in the low latitude ocean, so the tropics and the subtropics. And this makes sense because we know that this is where nitrogen is limiting. So I just want to focus on the South Indian Ocean because this is my study region for today's talk. And what you can see generally is there is kind of a widespread signal of N2 fixation predicted for the entire South Indian Ocean. So to investigate N2 fixation, there are multiple geochemical tools. I'm going to speak about two of them today. So the first is the nitrogen isotope ratio of nitrate. So we refer to that as delta N15. And the reason that this is a tool is because N2 fixation introduces a delta N15 that is really low. So its N member is around -1 per mole. and it's low relative to mean ocean nitrate delta N15, which is around or between 4 and 7 per mole. So the N member for interfixation, it's nitrogen isotope N member, is low, which makes it really easy to identify, relatively easy to identify, I should say. So in the thermocline, right, where the nitrogen fixes, so nitrogen fixation happens on the surface, so we produce organic matter, and then that organic matter needs to sink through the euphotic zone and be remineralized in the surface, remineralized in the thermocline. And that is where we can see the signal recorded in the nitrate pool. Okay, so we're looking at the nitrate, the DIN pool. Okay, the second geochemical tool is we can look at nutrient ratios. So here, I'm going to talk a bit today about N star. So the reason that it's useful is because N2 yields organic matter with a high N2P ratio. And this ranges considerably, but it ranges somewhere between 20 to 1 for an N2P, even up to 180 to 1. So that its N member in terms of N star is also significant. And how that manifests is we see a high thermocline N star, because again, the newly fixed organic matter that's generated in the surface needs to sink down through the euphotic zone and be remineralized into the nitrate and phosphate pool, and that's where we will see the signal. So I'll bring up these 2 points again later when I show some of our data. Okay, so this is a map showing thermocline circulation for the South Indian Ocean, where the white arrows are showing circulation pathways for, by thermocline, I mean it's about the 200-meter depth level. The map color is the dynamic height climatology, so it's indicating circulation of the thermocline, where the darker blue colors in the Southwest Indian Ocean. I hope you can see my mouse. Here, this just indicates tighter recirculation or retention of the waters in the Southwest Indian Ocean, and this is typical of Western intensification in all subtropical gyres. I'm also going to point out 3 data sets. So there's a pink data set that samples across the Agulhas Current and into the open ocean. I'm going to refer to this a lot today. And then we have 2 other isotope data sets, new data sets. This is the swings data set some people might be familiar with. And this is the IOE2 data set that was collected in the Mozambique Channel. Okay, so if we just focus on the Agulhas Current, it is the western boundary current for the South Indian Ocean, and it is one of the strongest, well, it is the strongest boundary current for its latitude in the world. And the system, the Southwest Indian Ocean, is characterized by strong heat and salt fluxes. And what we know about other western boundary current systems is that they also are characterized by nutrient streams, so strong lateral advection of nutrients into the system. But the biogeochemistry of this system is uncharacterized, so we don't have a good sense of what the nutrient fluxes look like. So when we started looking into nutrient supply mechanisms for the system, it required that we essentially build a biogeochemical framework for the region. So what I'm showing on the left is a map of the Indian Ocean basin, where we're showing water masses, their formation regions, which are the circles or the squares around the boxes, and then also how they are laterally advected into the system. So what you can see, what's immediately obvious, is that lateral advection is particularly important for this region. And on the right-hand side, I'm showing a TS plot where the color is nitrate concentration. So this is for the discrete data shown on this map here. And what I want to draw your attention to is above this 26.4 isopycnal, so roughly above about 500 meters, we see a strong differentiation in the thermocline and surface waters. And why we see that is that tropical waters are really different, both physically and or hydrographically and biogeochemically, to subtropical waters. So what I mean is that tropical waters are fresher, they are warmer, and they have higher nitrate concentrations, so the colors, relative to the subtropical waters. And if you look on the map, you can see how tropical in the red, how these waters are advected into the system, and the subtropical waters, how they are advected into the system. I just want to draw your attention again to this pink transect that samples across the Agulhas Current, because I'm going to show data, I think in the next slide. Yes. Okay. So what you're looking at here is the current speed from that transect that samples across the Agulhas Current. So on the left-hand side, you have the African margin, and you're moving southward as you move toward the right. This is the upper 500 meters. The color, so the red colors are basically indicating southwestward flow. So this is the Agulhas Current. I'm using this bracket at the top to indicate its reach. But this red contour that I'm using is the 1 meter per second contour, and that indicates the core of the current. And this will become, why this is important will become apparent just now. The white contours are indicating the isopycnal boundaries of the water masses, which we're just going to generally define here as surface thermocline and subazotic mode water. So this is the underlying source water. And these white circles are indicating the mixed layer depth. Okay, so what we already know about the Agulhas current and all western boundary currents is that there is a strong potential vorticity gradient that exists across the current. And what that does is it inhibits lateral mixing. So how that manifests is we see tropical waters constrained to the inshore side of the current, right, which I'm annotating on the top here, and subtropical waters are constrained to the offshore side of the current. So there is literally a divide of these two source waters, which I said are both different in salinity and some of their biogeochemical characteristics. So there is this divide. We already know that this exists, at least that the potential vorticity gradient exists and that it divides the water masses. But this work has made us think about how this impacts, how this impacts the biogeochemistry. So let's get into it. Here I'm showing a nitrate section plot from the same transect, obviously. So in the surface waters, you can see low nitrate concentrations near zero, and they increase with depth. And now I'm showing the nitrate delta N15. So what is immediately obvious is that in the surface, you have really high delta N15 values, so greater than 8 per mole. And this is typical of phytoplankton assimilation of nitrate. So phytoplankton will draw down the nitrate, right? That's why it's low in the surface. And in doing so, they will raise the delta N15, right? That's why it's really high. But what I want to draw your attention to is the thermocline. And what we see here is low delta N15 nitrate. And that is low relative to the underlying source. So here, delta N15 is between 4 and 5 or 4 and 6 per mole, so these purple colors, and subazotic mode water below it is about 6.9 per mole. So this signal is classic of N2 but I'm going to convince you in another way anyway. So what you're looking at here is the same data, but you're looking at density profiles. So on the left, I'm showing nitrate concentration. In the middle, nitrate delta N15, and on the right, N star. And the warm colors, so these pinky, maroony, orange colors, those indicate stations that sampled tropical waters. And these cool color stations, so the blue and the green, those are stations that sampled subtropical waters. Okay, and what you'll notice is that relative to the source, because it's always important to be relative to the source of right, what sets the supply, the thermocline nitrate delta N15 is lower. Okay, and again, when we look at an N-star space, we see that N-star is elevated in the thermocline relative to the source. And I showed you how we can use these geochemical traces as evidence for N2 fixation. We did, in the paper, assess a whole lot of other processes that can generate these signals, but we were able to conclude that the signal is local N2 fixation. Most of it is. So here I'm showing a map, that same map again, and I just want to make the point that the system is dominated by these strong lateral advection signals, right? These water masses are advected laterally into the system. So that means that we needed to consider lateral nutrient advection, or more specifically, how an N2 fixation might be laterally advected into the system. And that's important for accurately estimating an N2 fixation rate, because we could be overestimating a rate by attributing N2 fixation in this system as if, when it actually maybe occurred outside of the system. So we used a one-box model, which is what I'm showing you on the right. And the point is just that we used nutrient fluxes and a nitrogen isotope flux to estimate an N2 fixation rate. And that rate ranges between 7 and 25 teragrams of nitrogen per annum. And I just want to make this point that this estimate is for the greater Agulhas region, which we define in this work as this maroon contour region. So it excludes the Mozambique channel. And we see an N2 fixation signal in this Mozambique channel, and whether it's generated in situ or if it's transported, we can't say because we don't have enough available data upstream of that, but this rate excludes the Mozambique channel. So this rate is also the first observation-based rate for the South Indian Ocean. And when we consider it relative to models, it suggests that 30 to 95% of the basin-wide N2 fixation rate is occurring in the system. And I should make the point, although I'm sure this audience is well aware, there are very little data available for the South Indian Ocean, right? In terms of from nutrient data to isotope data, there is just not a lot of data. So these models are obviously extrapolating in this region because there aren't observations. Okay, so now I want to talk about the other nutrient supply mechanism that I introduced in the beginning, and that is upward mixing of deep nitrate. So we know that quantitatively, this flux of deep nitrate supply is dominated by winter mixing or convective mixing, seasonal convective mixing. But what we also know is that episodic entrainment events also play a role in supplying nitrogen to the surface ocean. So what I'm showing here is a global map of mean eddy kinetic energy. And it's just a proxy, well, I'm using it here as a proxy for turbulence or mixing potential. So here I have overlaid the broad scale subtropical gyre circulation, where the pink arrows are indicating the major western boundary current systems. And the take home that I want to make here is that western boundary current systems are characterized by high eddy kinetic energy. So where you see dark blue on the back of the map, you can, it's coincident with the pink arrows. But also that these high turbulent regions are coincident with strong surface nutrient gradients. So this maroon contour here all over is showing the 1 micromolar surface nitrate concentration. We know nitrogen is limiting in the low latitude ocean, so nitrate in the surface ocean decreases equatoid. So the point is that these western boundary current systems are located in regions where we have a high mixing potential or a lot of turbulence, and also where we have strong nutrient gradients. So that suggests that these systems should host significant sub-mesoscale vertical nutrient fluxes. And I'll talk about scales in a second. I'm going to talk about them right now. So identifying the impact of sub-meso or small-scale nutrient fluxes is difficult. And it's difficult for 2 primary reasons. More reasons, but I'm just going to talk about 2. So the first one is that the scales that these processes happen are small, right? So when we talk about mesoscale and sub-mesoscale, we're looking at spatial scales of 1 to 100 kilometers and temporal scales ranging from hours to months. So sampling these like ephemeral events is of course difficult, particularly with ship-based sampling. Okay, one of the second biggest challenges in tracing the sub-mesoscale vertical nutrient supply is that when you supply nutrients to the surface ocean, or at least a nitrogen-limited surface ocean, biology or phytoplankton are going to take up this nutrient and remove the tracer, right? So there are these overlapping biological processes that remove the tracer really quickly, right? They will assimilate the nitrate if they're nitrogen-limited within hours. So that makes it also difficult to quantify or even just assess the impact on productivity. So I'm going to introduce another tool, a geochemical tool. So it's nitrate delta 1518, and I'm introducing it because this tool can actually overcome the 2 challenges that I just identified. So nitrate delta 1518 is defined as the delta N15, so the nitrogen isotope ratio of nitrate, minus the delta O18. So this is just the oxygen isotope ratio of nitrate. But just think of this as a tool. It's a tracer. And there are processes that change delta 1518, and I'm not going to get into that too much today. But what is important is that there are processes that do not change nitrate delta 1518. And those processes, fortuitously, are the same processes that I identified as challenges. So the first one is that physical mixing of water parcels does not change nitrate delta 1518. So it is conserved during physical mixing. And the second one is that phytoplankton assimilation of nitrate, so the biological response to the nutrient supply event, also does not change nitrate delta 1518. So this makes this tracer quite a unique tool to assess sub-mesoscale nutrient supply. Okay, so I just want to make this point again. We can overcome the challenges that I identified. So if you had to upwell deep nitrate into a low nutrient surface water, the delta 1518 will record the source of the nitrogen or of the nitrate that was supplied, even as assimilation draws down, or phytoplankton assimilation draws down the nitrate to near 0 concentrations. You will still be able to record the source of that nitrate. So I'm going to show you the data to convince you. So here we are looking at nitrate delta 1518 across the Agulhas currents. This is the same transect that I showed earlier. And what we can see, okay, first let's just look at 3 layers. So at the bottom, we have subanodic mode water, which has a very high delta 1518 of around 3.4 per mol. And it's high because of partial assimilation in the Southern Ocean surface waters. Then we consider the thermocline layer where delta 1518 is lower than subnautic mode water. And this is because of N2 fixation, which I spoke about just now. So N2 fixation lowers delta N15 more than delta O18. It's not important why it does that. The point is it lowers delta 1518. Okay, and then in the surface layer, we have even lower delta 1518, and that is because at the base of the euphotic zone in this system, we observe coupled partial nitrate assimilation and nitrification, and the result of that is just that it lowers delta 1518 even further than the thermocline value. So just consider these three layers where delta 1518 is lower as you move to the surface. If you look at the arrow I made on the right, you can just consider these delta 1518 lowering as you rise up in the water column. So when we look at the delta 1518, now I've annotated on here, what we see is 3 incidences of high delta 1518 nitrate in the surface. Okay, so I've annotated 1, 2, and 3, and this reveals that deep nitrate has been injected into the surface layer. There's no other way to raise delta 1518 in the surface. So it shows us, it's tracking that deep nitrate has been injected into the surface. And why we care about where the nitrate comes from or what the sources of the nitrate is, of course, because it has implications for new production, right? If you are supplying nitrate from below the euphotic zone into the surface layer, you are fueling new production, which means you are increasing ocean fertility. So we use this data and some other biogeochemical and hydrographic data to try and characterize the sub-mesoscale dynamics that drove these nutrient supply mechanisms. So if we start at the first mechanism, so the number 1 here, what you're looking at is an anticyclonic eddy. So that's a downwelling eddy. And you can see high delta 1518 at the edges of the eddy. So there have been upward injections at the edges and at the core of the eddy, right? It's downwelling, so you downwell the core. So what we see is based on the delta 1518 that nitrate from about 200 meters has been injected at the edges of this anticyclone. And then if we look at the second incident, right on the shelf, which is about 52 meters depth, we see really high delta 1518 nitrate of about 3.4 per mol. And that is the same delta 1518 as subanautic mode water. So what that indicates is that subanautic mode water from about 400 meters deep has been upwelled onto the shelf and flushed the shelf. The entire profile has a delta 1518 of 3.4 per mol. And then the third mechanism that we see is located at the offshore edge of the current core. So that's why I made this contour here. And what we see is this high delta 1518 signal that's kind of wedged itself between this the mixed layer here as these white circles and the top of the thermocline. So this 25.5 isopycnol is the isopycnol for the upper boundary of the thermocline. And you can see this deep nitrate has been supplied in between, right into just below the mixed layer and the top of the thermocline, but you don't see it surface. So this is a subsurface entrainment event because on the surface above the mixed layer, we still see typical low delta 1518 or typical of surface waters. So I'm going to show some of the other data, biogeochemical data that helps support our ideas here. So what I'm showing now on the right is the nitrate concentration. So I've shown this before. And in a system that is nitrogen limited, like this one, and that is not light limited, like this one. So in the subtropics, you won't be light limited. Well, we do have PAR data, but you're not light limited over at least the upper 100 to 150 meters. So what that means is that if you supply nitrate, so the limiting nutrient, to the surface ocean, phytoplankton will rapidly assimilate this nitrate. And by rapidly, I mean within hours of supply. So it, of course, has something to do with the relative rates of supply and drawdown. But in general, elevated nitrate concentrations in the surface layer indicates us that the nitrate supply was ongoing. Okay, so if you look at the edges of the eddy, here we see elevated nitrate concentrations at both edges. So that indicates that this eddy edge upwelling was ongoing when we sampled the event. On the shelf, we also see very elevated nitrate concentrations. And what it shows is actually a combination of high nitrate or nitrate-rich subanautic mode water and the basically nitrate-free surface waters that were sitting here before. And if you look at the third mechanism, what we see here is actually low nitrate concentrations, right? So this purple color below the mixed layer. So that tells us that The delta 1518 tells us that we supplied deep nitrate, and the nitrate concentration is telling us that phytoplankton have already assimilated this deep nitrate. So that gives us the sense that this event happened probably, I don't know, on the order of days ago, maybe 1 to 3 days before we sampled it. Okay, so now I'm showing the AOU across the transect, and we know that AOU rises in the thermocline where you remineralize organic matter, which means you consume oxygen, so you raise AOU. And when you have an event that mixes thermocline water to the surface, what we know is that AOU or oxygen will very rapidly equilibrate with the overlying atmosphere, so within hours. So if you see high AOU from the thermocline mixed up or injected into the surface layer, and it's still maintaining some of that thermocline signal, it suggests that this event happened recently or is ongoing. So that's what we see at the edges of the eddy, more so at the offshore edge of the eddy, that we see this slightly higher AOU signal, which tells us that this event happened, or at least this event is ongoing, because we still see a little bit of a thermocline signal. On the shelf, we similarly see the same thing. So we still see this high AOU signal on the shelf, which means it hasn't yet equilibrated with the atmosphere, which means the event must have been ongoing or extremely recent. But when we look at the third mechanism, what we see is this low AOU signal below the mix layer, right? So typically we expect AOU to increase immediately below the mix layer, where it does everywhere else in the transect, because that is where you start to remineralize organic matter and consume oxygen. So this low AOU signal below the mix layer and between, right, the base of the top of the thermocline, what this indicates is that there has been recent ventilation below the mix layer, right? So the waters below the mix layer have recently been in equilibration with the atmosphere and have mixed. Okay, now I'm showing the salinity section from the transect, and I'm not going to talk here about mechanism 1 and 2. I just want to draw your attention to mechanism 3. So what we see, and I've contoured it in white here, is this arm or this tongue of high salinity, high density surface water subducting beneath a lower density or lower salinity surface water. So this subduction of the waters below the mix layer is indicating recent or somewhat ongoing, but recent sub-mesoscale overturning, right? We can really see the overturning cell in the salinity data. Okay, so now I'm showing the current speed, which you've seen already. So if we look at the edge of the eddy, or at least at the location of the entire eddy, we can see the two halves of the eddy field, right? So coming southwestward component and the northeastward component, which is pretty strong here at the offshore edge of the current, offshore edge of the eddy. Over the shelf, we don't have broadband data over the shelf, so we can't really make any conclusions about that. And what we see in the region of the third mechanism is all we can really see is that there are strong horizontal velocity gradients that you can see, and also strong vertical velocity gradients. So if you look carefully, for example, at 150 meters, the velocity is actually a little bit stronger than at, let's say, 80 meters. So there are really strong shears in the velocity data. Okay, so here I'm showing the inferred vertical velocity data. So what we did is we took the depth integration of horizontal convergence from the shipboard ADCP data, and regions where we have positive vertical velocity is upwelling. So the green colors indicate upwelling. So if you look at the eddy edges, what we see is generally upwelling at the edges of the eddy. So this is consistent, right, with the shoal to mix layer. At the core of the eddy, we see this red color, which is downwelling, right? And this is typical of an anticyclone, downwelling at its core. Okay, so we don't have data over the shelf, but the near slope velocities that we do see here are green, right? Which means they're positive, which indicates upwelling. So that's consistent with this inshore upwelling mechanism that we see. And then again, here at mechanism 3, right, we can't say anything absolutely from this data, but what we do see is that there are strong horizontal gradients in the vertical velocity, right? So you can see it goes green, red, green, red. So strong horizontal and vertical gradients or shears in the data. And what we know is that in winter, across the Agulhas Current, there are these strong density, so I didn't show the density data, but there are strong density gradients and velocity gradients which I have shown. And what these high shear regions do is it primes the system for baroclinic and symmetric instabilities, okay? And what these instabilities do is they can generate vertical recirculation cells that can actually reach below the mix layer and mine nitrate from the thermocline and actually inject it upwards into the euphotic zone. Okay, so there is a mechanism at really small scales, so at sub-meso scales, that generates vertical velocities that can entrain nitrate and inject it into the euphotic zone for phytoplankton then to assimilate. So this is this is what we propose is happening, that this high shear zone is generating enough of a vertical velocity that we can actually entrain nitrate and phytoplankton have assimilated it. So obviously evidence for dynamics at the scale is scarce. It's very difficult to attain. So this tool, this Delta 1518 tool, is really provides really unique insight into the vertical nutrient supply and then also the impact on productivity. So this is a schematic that uses Delta 1518 to highlight the 4 nutrient fluxes I spoke about today. So N2 fixation and the 3 vertical nutrient fluxes. And I guess just to conclude, right, I spoke about the N2 fixation rate that we derived for the greater Agulhas region. And we know now that the dynamics of the Agulhas current enhance vertical nutrient entrainment in the surface waters, and all 4 of these nutrient flux mechanisms increase regional productivity. And what we know is that all western boundary current systems are characterized by this high eddy kinetic energy, right? I showed that on the map, which means they're all turbulent systems at these scales. So that suggests that quantitatively, western boundary current systems or western boundary current induced nitrate supply is probably significant. And with the prediction that subtropical gyres are stratifying and that their productivity will decrease, there is a chance that the western boundary current induced nutrient supply mechanism may contribute to offsetting the decline in subtropical gyre productivity. So unfortunately, with the data that we collected from this cruise, we were not able to quantify these nitrogen fluxes, but given how significant we think they could be, we do intend to do so in the future. So I would just like to make some of these acknowledgements, and if anyone has any questions, please go ahead and ask. Thank you.
Thanks a lot, Tanya, for explicitly explaining such intricate reality of ocean. I now invite viewers to ask questions. We have a few questions in our chat box. First question is, have you looked at how changes in the Southwest Indian Ocean during ENSO events influence nitrogen fixation?
Yes and no, it's okay. I get it. Yes and no. No, because we don't have enough data to try and look at something, over that kind of scale of variability. I would like to, but we have recently collected some more data from this region. So hopefully in the near future, we can maybe start looking at those kinds of processes. And then the yes aspect is that I am collaborating with a co-author in Germany at MPI, who is looking more on a paleo time scale, but the impact of these kinds of climatic scale variability on N2 fixation in the basin. So not me personally, but I am aware that some of this work is starting to be done.
Next question is, with the broadening or warming of the Agulhas current, how do you expect the nitrate concentrations to change in the future?
Good question. Okay, so we have thought about this a bit, and I guess the conclusion from that Beal et al. Paper, right, about broadening and warming of the current, they also talk about how the mesoscale variability is increasing. So what that means is that, so I spoke about how the potential vorticity gradient is inhibiting lateral mixing, but this gradient, the increased meandering, so the current sometimes meanders, shifts off the coast and back. This kind of meandering will increase this cross-shore exchange. It'll basically erode that potential vorticity gradient, which means that the tropical waters and the subtropical waters will actually be able to mix. So increasing that kind of lateral scale or cross-shelf exchange has probably meaningful implications for productivity. I'm not sure what it would mean for N2 fixation. We typically think about N2 fixation or recording that signal of a thermocline timescale, so like 4 years maybe. I don't know how that's going to impact it, but what we think will happen is that as the system warms and kind of intensifies, the mixing is going to intensify. So perhaps these vertical supply mechanisms will increase. But yeah, I think we could probably do a little bit more and collecting more observations to have a better sense of how the system might change in future.
Next question is the IIOE2 dataset in the Mozambique channel freely available?
Yes. So the isotope data and the nutrient data are freely available. You can find the link. Wait, if I go back one, I don't know if you can still see my screen. I think you can. If you look at the QR code in this paper, we provide that data in the Zenodo database. You can also email me for it, but it is available there. And I believe that from that cruise, there were more samples collected on the Western Shelf. We just only have access to what we have access to.
Okay. Next question we have from Mar Benavides. She's saying, wonderful talk, thanks. Her question is, which mechanisms can make the WBC fuel nitrogen availability in the subtropical gyre in a future stratified ocean scenario? Seems that the lateral transport is minimal, at least to reach the central part of the gyre.
Right. So I think, yeah, when I make that comment, what I'm more referring to probably is like low latitude decline in productivity. So the subtropical gyres are expected to expand, so will probably reach further into these western boundary current systems. So I guess in that sense, the western boundary current system productivity may decline. But maybe combating that, like I said about this warming not expanding current, is that the mixing in these systems might intensify. So perhaps not. I don't know. I do think as a low latitude system, the western boundary current could offset some subtropical gyre productivity. But if we're just thinking of a local impact, maybe even stratification of western boundary current systems, right? If that's one way that we decrease productivity, maybe we can offset it by this increased mixing.
Next question is from Sudhish. He's asking how the nutrient stoichiometry is varying in these regions. Do you see any nutrient limitation, phosphorus or nitrogen?
Great question. Thank you. So I didn't have time to get into nutrient limitation, but I have personally spent a lot of time thinking about it. So what we know, if you just plot like a surface map of P star, we can see that the whole Indian Ocean is nitrogen limited in the sense that there is a P excess, so positive P star across the whole ocean basin. And that is largely derived from the denitrification in the Arabian Sea. So, right, we remove N and not P, and we can see that signal, even in the surface data, permeate from the Arabian Sea southward, and it's kind of, transported laterally down the western boundary, well, across the basin, but particularly down the western boundary. So I think in general, the whole basin is nitrogen limited, but I think, again, we need far more data to be able to make that, to validate that statement. There just is really little data available for the South Indian Ocean.
I think next question is from Matthew. Matthew, you can, you haven't put your question on chat. Maybe you can ask directly.
Yeah, sorry. Thanks, Tanya. That was really, really cool talk. And that data set is really cool, the transect. I was just wondering, do you have is it just 1 year or one snapshot, or do you have multiple years of that transection? And can you do like an interannual comparison?
Great question. So this data is from 2016, but we did collect data in 2018. And we recently collected now in 2023 a really beautiful sample set because we haven't analyzed it yet, which I think is going to be really promising, right? We thought about the lessons we've learned and what kind of things we want to look at and the scale we want to look at them. So I think that data set will be pretty insightful. In terms of interannual variability, we don't expect, right, for an interfixation signal that's generating over 4 years, residence time of a thermocline, we don't expect much interannual variability. But, when processes like these, climate scale variations, so Indian Ocean oscillation and all that, you may expect differences between years, how meaningful, how quantitative that is, how quantitatively meaningful that is, you know, year in and year out, I'm not sure. But what I think would be really interesting and what is missing in this region is some kind of estimate of lateral nutrient transport. So for example, for the Gulf Stream and the Agulhas Current, we have a really good sense of how much nitrate, for example, is being laterally evicted. Like we've quantified that. And while I try to do that on the box model, right, it's based on a lot of assumptions. So it would be really great to have a sense of what the nutrient flux looked like. So matching the transport, right, like water volume transport of the Agulhas Current with the nutrient concentrations, because I think that has strong potential to vary annually, which then does have implications for productivity and, right, all the fishery systems that are thriving on the Agulhas Bank. I think it's important work to be done. We just need someone to do it, I guess.
Thanks. Yeah, it's super interesting.
So we have no more questions further. So thanks a lot, Tanya. And I hope we'll see you all on next Friday of first Friday of October month. With this, we close this session. Thank you.
Thank you so much, everyone. Thank you for the opportunity to talk. I really enjoy sharing this work, so thank you.